So in the cell, both strands of DNA are replicated at the same time. So these two strands, they start replication at the same time. So it requires separation of the two strands of the double helix to create two template DNAs. So as you know, one DNA molecule is present, and in this one molecule two strands are helically coiled. So when replication will start, it will start not only on one strand but on both the strands. So it is compulsory that these two strands must be opened and must be separated to start replication on these two strands. The junction between the newly separated template strands and the unreplicated duplex DNA is known as the replication fork. So replication fork is the junction between a double-stranded DNA and the single-stranded DNA strand. So here you can see in this diagram, this is a double-stranded DNA, and when it will replicate, it will open, it will bifurcate, and this is the junction between this single-stranded DNA molecules and the double-stranded DNA molecules. This junction is called replication fork. So replication fork initiates the DNA replication. The replication fork moves continuously towards the duplex region of the unreplicated DNA. So as you know, this is a duplex. It opens and it makes the replication fork, and this fork start moving towards the unreplicated DNA molecule, that is double-stranded DNA molecules. It keep on moving, and the two strands keep on opening up. As the fork moves, it creates two single-stranded DNA that each act as a template and direct the synthesis of a complementary strand. So at a replication fork, two single-stranded templates are formed, and these two templates make two new strands. So in this way, a single DNA molecule will be replicated into two DNA molecules. The anti-parallel nature of the DNA creates a complication for the simultaneous replication of the two exposed templates at the replication fork. You know, DNA, the two strands of DNA, they have anti-parallel nature. One strand runs from 5 to 3 prime, and the other strand runs to from 5 to 3 prime. So in one side, one strand has 5 prime, the other one has 3 prime. On the other side, one strand has 3 prime and the second strand has 5 primes. This is called anti-parallel arrangement of the strands. So this anti-parallel arrangement, it creates some complications for the synthesis of new strands on the two single-stranded templates. Because you know, DNA is only synthesized by elongating a 3 end, that is, this synthesis only from 5 to 3 prime. Only one of the exposed templates can be replicated continuously as the replication fork moves. So when two strands open, these two strands have opposite arrangement. One has 5 to 3, the other one has 3 to 5. So only 5 to 3 strand will be synthesized continuously as the fork moves. So synthesis at this strand keeps on as far as the fork is opening up. The newly synthesized DNA strand directed by this template, that is 5 to 3 direction, this newly synthesized strand is called leading strand. So the new strand which is synthesized along this template, this new strand is called leading strand. As you can see in this diagram, This is the replication fork, and this is 5 prime, and this is 3 prime. So new strand is synthesized here. This is new strand, and the new strand will have a 5 prime on this side and 3 prime on this side. So this synthesis will be continuous and will continue until this fork will move towards the duplex side. But here on the other strand, this synthesis is not possible due to reverse direction of the strand or template. Synthesis of the DNA, a new DNA strand directed by the other single-stranded template is more complicated. The one you have seen in the previous photo, you have seen that the synthesis is continuous, while on the other strand the synthesis is very complicated. This template directs the DNA polymerase to move in the opposite direction of the replication fork, because the direction, the 5 to 3 prime direction on this template will be opposite to the fork. Fork will move towards this direction and synthesis will move towards this direction. The new DNA strand directed by this template is known as the lagging strand. So on this template, this new synthesis, new strand is called lagging strand. This strand of DNA must be synthesized in discontinuous fashion. It cannot be synthesized continuously. Synthesis of the lagging strand must wait for the movement of the replication fork to expose a substantial length of template before it can be replicated. So when a replication fork moves, the two strands open. In the one strand, a leading strand will be synthesized, while in the other template, lagging strand will be synthesized, and this synthesis will occur in fragments. So when the fork will move forward, it will expose substantial length on this strand, and then the new fragment will be synthesized. Each time a substantial length of the template is exposed, DNA synthesis is initiated and continues until it reaches the 5 prime end of the previous newly synthesized fragment of the lagging strand. So as you know, in this strand the fragments are synthesized. So when the replication fork will open, this length will be exposed. Synthesis will start from here and will move towards this until it reaches the 5 prime end of the already synthesized fragment, and so it continues like this. The resulting short fragments of new DNA formed on the lagging strand are called Okazaki fragments. These Okazaki fragments, they vary in length. In prokaryotes, they range in length from 1000 to 2000 nucleotides, while in case of eukaryotes, the length of these Okazaki fragments is 100 to 400 nucleotides. So as you know, this is the double helical DNA. It opens up, and when it opens, this is you can see this is a leading strand. A new strand is synthesized in this direction, that is from 5 to 3 prime, while in case of this strand, the synthesis occurs in fragments, and the direction of synthesis is away from the fork. This is 5 to 3 prime. So these fragments are called Okazaki fragments, and after synthesis these fragments are joined. Shortly after being synthesized, Okazaki fragments are covalently joined together to generate a continuous intact strand of new DNA. Okazaki fragments are therefore transient intermediates in DNA replication.